गर्म पानी दे दो मैंने पी लूं Oopsie. So welcome class for third session we are having maybe. So earlier I was taking your classes over the Zoom, which goes for like 40 minutes and then it stops and I have to start again. So which leads to production of two videos. One is of 80 MB, one is for 60 MB, 86 or something. And I spent like 3 to 4 hours in order to make them small so that I can share with them with you, but it was not possible. So that's why I switched to live streaming, because in this it will be recorded over the YouTube. You can watch it anytime, anywhere, wherever you want. You don't have to save them in your laptop, and they will be available there over my channel. So please do subscribe to my channel, whosoever are present. It will really help me. I had so far like 846 subscribers. So I'm just looking for around 150 subscriber more. Then my channel will start. I can also appreciate others how to initiate your own YouTube channel, how you could make some educational videos, how I'm using OBS software. You can ask me personally anytime. So I will be happy to share with you also how things we do online things. So you're most welcome to ask me anything. I will be happy to grow everyone, because I don't want to grow by myself if I'm growing in the field of education. If somebody wants to learn how we can have our own educational channel and you can teach and get a lot of people on your platform and making them whatever you have the knowledge, how we can share with them. So this is the best platform that ever have been created. So feel free. So I will not waste much time, because these classes are only for 1 hour. So we have already wasted over 10 minutes. On that note, I will start with the transcription that we left last time. So, very basic, very gentle. So transcription is basically a process of creating your RNA copy from the sequence of DNA. So transcription is the first step leading to the gene expression. So in DNA is making your RNA in the field of transcription, but in the presence of reverse transcription, like an enzyme reverse transcriptase, it could produce DNA also. So that thing is also quite possible. Yeah. So during transcription, a DNA sequence is read by RNA polymerase, which produces complementary and triple RNA strand, and instead of thymine, we use uracil as a component part here. In order to understand this transcription and translation process, I will not go through this sequence. I will be happy to share. Equivalent means equal, bache, like almost similar. So I will take you a very simple video about transcription and translation.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life.
So students, have we done this video before, or because I'm going through a lot of things at the moment. We have a college in the morning. I'm going through some multiple workshops, so sometimes I forgot on which topic I was. Did I showed you this video before, or we are done with that video already? Because if we are done, then I don't want to do it again. Anyone could please give their comments on it? We have okay, fine. So it's not required to do it again. Cool. So in this transcription process, it's mainly your proteins which have to be transcribed. The results of the transcriptions are the messenger RNA. So the DNA is read from 3 to 5 prime end during this process in the presence of your RNA polymerase, and complementary RNA is created from the direction 5 prime to 3 prime end. Yeah, and only one of the two DNA strands called the template strand is used for the transcription process, because RNA is usually single stranded. So only one template is used. RNA polymerase will move from 5 prime to 3 prime end, and at the end you will have a single mRNA being produced, where the other strand is called as coding strand, and others is a single strand. So that's how you have your mRNA production. And also in the mRNA production you have sorry, exon 1, exon 2, exon 3, and in between their introns are present. And during the transcription process, RNA splicing is done, where all your introns will be removed out, kicked out, and mainly your messenger RNA having your exons will be there during the RNA synthesis and processing. And also one more aspect of this RNA processing is a reverse transcription, which is done in the presence of a reverse transcriptase enzyme. And also in this, the DNA copies are made via your new RNA, and beyond that, retrotransposons also spread by copying DNA and RNA from one another in this case. So as general, we have discussed this central dogma of life, where your DNA is producing RNA and then protein, and RNA could also self-replicate or then go back to also to DNA, and DNA could also self-replicate. So these are the blue ones are the general method, and the red ones are the special ones which happens inside the cell. Then the translation, which is the first stage of a protein biosynthesis. So in this, mRNA is produced by transcription, decoded by the ribosomes to produce specific amino acid chains or polypeptides, then which later take the shape of a quaternary structure of a protein. And the translation that happens all happens in the cytoplasm, where the large and small subunit of your ribosome, your 70S and 30S, are located and bind to your mRNA with the help of your tRNA to produce your protein. So this was a process of translation, where you can see your messenger RNA is being there, and on top your ribosome came with the help of your tRNA. It started with the start codon of methionine, and then comes the next tRNA, and then the next amino acid being attached to it, and here they get attached to your anticodons of your tRNA, and then this way the polypeptide chain of amino acids being keep going on increasing, and at then by the stop codon it will stop and your translation will be stopped at that point. So basically it has 3 steps: initiation, elongation, and termination, and by the end of that you have full-fledged your protein which get folds and make things done. I think we did that part. I have a slight memory of it. Then comes your genetic code, in which you write on the first base UCAG on the left side, and then UCAG on the second base for there on the top, and on the right hand side you write the 4 letters UCAG like this. And then what you do is you take the first letter from the first base, second letter from the second base, and third letter from the third base. And like this you will have 4 bases, UUU, UUC, UUA, UUG. So this will be your phenylalanine and this will be your leucine. Yeah, and on this side these all categories will be called as serine. Here the all categories will be called as proline, threonine, alanine, valine, leucine, arginine. So you can remember at least these boxes, big boxes, having full-fledged names. Because sometimes you might be asked give the 4 genetic codes of proline. So it's very hard to remember. Even for me it's very hard to remember, but what I do is I just create a very fast in my mind these tables on the notebook, UCAG, and then write them, and then I remember that this is on this side is serine, then on the bottom is proline, then threonine, alanine, on the left side is leucine, valine, and then you can start the arginine also and glycine, and then the rest of the boxes having 2 parts. Here in the top right 2 corners will have your stop codons UAA, UAG, UGA, and on the left third part bottom here you will have methionine as a start codon AUG, and the rest is isoleucine. So it's you can do your own, find your own tricks and trips in order to memorize all these parts. So what is genome? Next part is we are done with the basics of your molecular biology. Here we have discussed how gene is acting as a hereditary unit, how DNA, RNA are being separated from each other, how many, and we have discussed various types of your RNA, messenger RNA, ribosomal RNA, tRNA. Then we discussed its various process of a central dogma with transcription, translation in details, and now we are going to discuss a little bit about genome. Genome is a entirety of an organism's hereditary information, so it includes all the information. It's like, you remember in the last in 90s from in 2000 years, from 1995 to 2000, we used to have telephone directory, the yellow pages we used to call them. So it used to have a particular of West Delhi phone numbers of everyone present with their name and telephone number in that directory. So it's the same like genome. So genome has all the details of a genes. Everything is there encoded for the DNA and RNA. Everything is being included in these genomes. Coding sequence, non-coding sequence, all the information of your amino acids, your proteins, everything is being decoded in that part. So that's acting like a telephone. That's, I found, the best analogy I could get it. And the best reason to understand this genome is to understand by comparing between different organisms. So let's say we have Homo sapiens mouse. So these are all the various models that we use to do in the genetic study or molecular biology studies for any disease resistance plants. I mean, some of you might be doing work on the plants in the field of I did that work in DRDO in Delhi related to sea buckthorn. So I was checking, what is this? I did very in 2008 I did that in there, and I was working with the sea buckthorn plant, which is grown in the higher altitude Himalayas. So these we took the fruits and leaves of that plant in order to check its antioxidant properties. Phenolic acids. Yeah, I'm remembering some of them. Alkaloids. All these phytochemical properties. So for that, as a model, if you're working on the plants, Arabidopsis thaliana is also used in that case. And then for the animal study, we also use your roundworms. And further, from the microbial point of view, yeast is one of the best model to study various diseases also. And then comes your bacteria, and then the yeah, two types of bacterias, E. coli and H. influenzae. So what you have to see, the best part here is that you have 46 chromosomes present in Homo sapiens and around 40 in the mouse, and on the other hand, the yeast has around 32, and whereas the bacteria has only one, which comes into your prokaryotes. So, and beyond that, the roundworm has 12 pair of chromosomes, chromosome number, sorry, and the Rhabdopsis thaliana is 10, and for the genetics, your fruit fly is 8, and the mouse is quite similar, that is 40. So whenever you want to study about any Alzheimer or Parkinson, any neurodegenerative diseases, or or other intestinal or any heart disease, cardiovascular diseases, anything that you want to study, so you can use these models, and these models are quite similar, their size is quite similar. So you can see that bacteria has the size base pair of 4.6 million, and whereas the Homo sapiens has 3.2 billions. On the other side, the genetics favorite model so far is the fruit fly, which has 137 millions of your base pairs of a genome. So you can see how everything is quite similar, and how things are quite drastic also in this page. So why we have to study the genome analysis? Genome analysis is done in order to do the prediction of genes in uncharacterized genomes and sequence, and to obtain the complete sequence of as many as genomes as possible, and it helps to understand the various genetic modifications, and also genetic modifications to develop new varieties at faster rate, like Bt cotton and Bt brinjal. So these are the transgenic crops that we have produced with the help of adding this Bt gene into them, which helps to fight out the disease, these insects that they eat. But somehow they got mutant against this brinjal, Bt brinjal and Bt cotton, and after getting mutant, they ate all the crops finally. So yeah, I want to discuss one point here from the transgenic point of view, that I was when I was in Germany, I was working with the transgenic crops with day and night. But when I put these crops, the GMOs crop, in the field for trial, so what happened? All the localites people came and they start to burn the field. Yeah, they within night they burn our all experimental fields, and the next day they were revolting that we don't want to eat these GMO crops, we don't want these experiments to happen, because they are not good for our future generation. Even if we eat them at the moment, which is likely in the market, if you go to your local market of vegetables or fruits, it is full of GMOs at the moment, nothing is organic there. So they might likely to change the upcoming generation. We might have defect at the genetics level, because what we are eating is genetically modified, so this might be change in the human body also. So based on that, I was young, I was very young and got influenced very badly at that time. So I switched off to the human medicine studies, and I did my lot of work in the hospital on getting heart failure to be cured, from the basic science point of view. But on nonetheless, so Europeans didn't want to eat, but on the other hand, the states, USA, they love GMOs. Actually, it's your Monsanto, Syngenta, Bayer crops, Bayer science crops. So many big companies are there, and they're having full-fledged business marketing there, and most of the, like, it has covered most of the the farming facility there also at a big level. So it's your view, it's your perspective. We want to eat them or not. These transgenic crops, it's up to your willing thing. So that was something about molecular biology, what we have studied. Now I want to just touch a little by little about the various tools that we use in the molecular biology, so that we are aware that what we're going to study in upcoming days from the protein perspective, from the molecular biology perspective, from the recombinant technology perspective, from the electrophoresis point of perspective, PCR perspective. So all these techniques will be touched little by little here. So let's start with first the gel electrophoresis, in which the basics principle is that DNA or RNA or proteins can be separated by means of electric field. So your DNA is actually electrically negatively charged because of the presence of the phosphate group in that. So whenever there is a negative electrode and positive electrode, the electric current has been flown within the gel, so your DNA will flow from negative to positive end. And at the end, after exposing them under the UV visible light, so we can check our DNA base pairs after exposing them, and we can say that this might be the whatever the readout that you're looking for, we can see it actually. Further, in the agarose gel electrophoresis, DNA and RNA can be separated on the basis of size by running the DNA through the agarose gel, and the proteins can be separated on the SDS base, the sodium dodecyl sulfate, in which SDS covers out your protein, and then your separation is done based on the electric charge and size. And this is sometimes you also use 2D gel electrophoresis, which is done in the field of proteomics. Which is a quite big field and quite extensively studied at the moment, because the people who are doing research over the mRNAs and DNAs, they are at the front level workers. But if people are working at the proteomics level, so you are working at the protein level, so that is the at the end that is the end product of your central dogma. So if you could identify your things at the protein level, so you can compensate and say that what is happening at the initial level. So all your DNA and RNA might be defected at that point also. So in that case, it's very helpful 2D gel electrophoresis. So I will explain its application soon, how we do that. Further, polymerase chain reaction. This is the polymerase chain reaction, which is an extremely versatile technique. I don't remember the scientist's name. What was his name? Kubel. He got the Nobel Prize winner for discovery of this PCR actually. I forgot his name. Oh no, how can I? Anyhow, sometimes we tend to forget things. It's okay. Further, polymerase chain is extremely versatile technique. So where we have a particular template that you want to increase size in million copies. Let's say somebody did a crime and we got the hair particle of that person and we extracted the DNA out of it, but the DNA that we extracted was in very low amount, just with one hair. So in that case your PCR might help. So that is one of the application of it. And also nowadays your RT-PCR, so there are two types of PCR. There is a real-time PCR and there is a reverse transcription PCR. So don't get confused between these two. So one is done for the quantitative real time. It gives you the results, the real time how much of your DNA amplification is done per cycle. Whereas the reverse transcription RT-PCR is a thing in which your mRNA from your messenger RNA, you produce a complementary DNA with the help of reverse transcriptase enzyme. So both are different, a bit different from each other, but this is, we usually get confused with them actually. So don't get confused with both terms. But in upcoming days, I will teach you all these techniques with proper videos, and how you can prepare the sample also by the professional companies that I have arranged them for you. So we will see how things are. So this is the PCR analysis is done, in which you have your three processes, mainly your denaturation, then annealing, DAE, then elongation. So in denaturation, first of all we separate up your template DNA from each other, by 94 degrees Celsius, around 90. And then you do the annealing. Your reverse primer and forward primer will come and attach to your separated template DNA. Then the Taq polymerase and your DNTPs will be come and play a role in the elongation process. The second process after for joining of your forward and reverse primer is annealing, in which its temperature is around 60 or 50 degrees Celsius. Then around 70 degrees Celsius your elongation will happen by addition of your DNA Taq polymerase and DNTPs. So they will make this full template. So by that cycle is over of your one complete denaturation, annealing and elongation, you will have 4 copies, and the same cycle will repeat again, the same temperature of 94, 50 to 60, then 70. So by this you will have 8 copies, 16 copies, 32 copies. By the 35th cycle you will have 68 billion copies. So that's the beauty of this PCR. So that's how the main principle is behind this, your DNA replications within the thermal cycler. And that's how it's so important, this technique is, and it has revolutionized your molecular biology field also. So earlier, because we had, if you're doing experiments in the lab, if you have taken one plant or 2 plants, and from the petiole you want to extract DNA, but you will get maybe in microgram, very less amount. But in order to do experiment, you need DNA at least in nanograms actually. So no, in milligrams at least, in huge amount you need. So in that case, yeah, this technique really helps. And the primer that you wanna make for that, and usually you prepare them with the help of a software where you check the annealing temperature, everything, and then the forward and reverse primers are made. Then you send them to the company to be prepared for you, and then you get them, and then you check that whether they are working or not by running over this machine. Then you get the results out of it. So some of the applications of PCR, that is the common application of PCR is a study of pattern of gene expression, and the task of DNA sequencing can also be assisted by PCR. And PCR has numerous applications to more traditional process of DNA cloning. Further, it helps in the patterns of genetic mapping, and PCR can also be used in parental testing, where an individual is matched with the close relatives also. Then comes your southern blotting, your blotting techniques, southern, northern, western, all these techniques in order to check your particular DNA. Southern is done for DNA, northern is done for RNA, and for the western we do the protein. To identify protein, we do the western blotting. So this is the general method which we do here with the genomic DNA. First we stick them with the help of endonuclease enzyme. You make a small patterns of it. Then you run over the gross gel electrophoresis with the high molecular weight at the bottom at the low molecular weight. Then we transfer this membrane with the help of capillary method over the nitrocellulose membrane or nylon filter. Then your gel, your protein, your DNA will be transferred. DNA, RNA or protein, whatever the technique that you're working, they will be transferred to the nylon membrane, and then you probe them with the radioactive phosphorus 32. Then if it will hybridize to the concerned DNA if it is present there, then we can expose to the X-ray film in order to identify it. So we will do also that technique in detail later part on the 7th day of your workshop on the topic called blotting techniques. So I will not go in detail here. So just give you a general overview about them, southern, northern, western. Then comes your molecular markers, which play quite important role nowadays in the market. In the Gujarat, a lot of big big market is there actually for the molecular markers, because these are markers. If you identify one a really good marker which could work for your what do you say, before I go ahead, I want to see if you have any questions, because I'm going quite fast I think for you. Yeah, Carey Muller is inventor. You're right. You're right. Fine, fine. So molecular markers, like there's a um, like there's a salt tolerance marker, there's a drought tolerance marker, there's a high yield marker, there is a insecticide uh, protective marker, insect protective marker. So there are many markers which could be present over the same gene, so we can use that for multiple functions. So in that way you have RFLP, restriction fragment length polymorphism, AFLP, your amplified fragment length polymorphism, RAPDs, random amplification polymorphic DNA, VNTRs, variable number tandem repeats, microsatellites, SSR, simple sequence repeats, SNP, single nucleotide polymorphism, STR, short tandem repeats, single feature polymorphism. So all these are quite, their terms are quite important. First of all, each term, their full form are quite important, and each one of them is different from each other. They are not similar to any of them. And further, it has its own applications. For example, first of all, there should be some characteristics of these suitable molecular markers. They should be polymorphic, yeah, codominantly should be inherited, and they should be frequently distributed throughout the genome. They should be present most of the time in the genome. They should be easy and cheap to detect, not very hard, and the results should be also reproducible. That whatever the things that you're working, they should be repeated nicely. So molecular markers can be used for several different applications. For example, we can use it for germ plasm characterizations, genetic diagnostics, study of genome, veterinary testings in the crime scenes, or the genomic response to selection in the livestock, in your cows, buffaloes, in that case. So first is your RFLP, which is a restriction fragment length polymorphism, which involves fragmenting a sample of DNA by restriction enzyme and can be recognized and cut wherever the specific short sequence occurs. So in this, first we cut the DNA from specific part where we are interested, and then we use it for the fragment for the individual and can be used for genetic analysis. Then AFLP, it's a amplified fragment length polymorphism. So we have the particular restricted fragment length and we increase its size to 1000 folds and can be used for comparison between closely related species also. Then random amplifications, in this we use PCR to randomly increase the particular segment of DNA, and then we can use it for the further function. Then there's a microsatellite polymorphism, SSRs, or simple sequence repeats, in which your particular sequence, let's say A, T, C, G, T, of 6 base pairs, they're being repeated again and again, again and again, again and again for particular function. Then there's a single nucleotide polymorphism, SNPs, which has a change in the single base pair. So if you can see, if there is a TA, then instead of TA, GC will be there. So which will bring a lot of change in your species. Your pairing of chromosomes will be different and leads to your having a different breeding program, crops or livestock. Then there is STRs. These are the short tandem repeats in DNA, which when pattern of 2 or more nucleotides are repeated and the sequence are directly adjacent to each other, and the patterns can be ranged in length from 2 to 16 base pairs. For like CATG in the genomic region, it will be typically in the non-coding intron region. So we use them for the forensic cases or genetic fingerprinting of individuals. Then we are done with all these molecular markers. So what is retrotransposons? So bache, these are the class 1 transposable elements. They're typically genetic component that copy and paste themselves into the genetic locations So mainly they are very, I mean, quite unique group of transposable elements and form large portions of genomes of many eukaryotes. That's something related to that. Yeah. Okay. Good. So my task is to at least have this unpending PPT since so many days we have started. Um, let's have done with this today at least. So the sources of DNA, for the isolation of your DNA, you can use your blood, buccal cells, cultured cells, bacteria, forensic samples, and all these. So in order to isolate DNA, I used to teach this with the virtual workshops before, but somehow due to the change in the um, you know Adobe Photoshop policies, it's not working. Or maybe they have funding problem. I don't know. Okay. Actually, earlier when we were having these classes, we used to have DNA extraction, gel electrophoresis, flow cytometry, PCR, DNA microarray. So I can share these labs with you. I don't know, maybe they were saying that they will open it up in the April end, but somehow, who's asking me about workshop certificate? Ah, it's the from the recent. Yeah, so maybe when it works, just please keep checking them. They're pretty awesome actually. They explain each things in very virtual platform, in very understandable manner. But sorry, it's not working. So we have to continue what we have with. So what you do is basically in the DNA isolation, three parts are the essential. That is your cell disruption, isolation of DNA, and precipitating the DNA. So these three different parts are one of the important while you're isolating your DNA. And in the cell disruption, this is mainly done by sonicating or grinding the samples. And we also have detergent in order to remove the membrane lipids. Then DNA is isolated by adding a protease if it is required. Then you precipitate the DNA in the presence of alcohol because it is insoluble in that. And then further we remove these alcohol soluble salts from it. Then we have a palette by centrifuging it, and then it's good to go. So here you can see that you have cell lysis and protein degradation enzyme proteinase K, and then you run it over the centrifugation, then you wash it with wash buffer, then you elute it again in the bottom, and then you have pure genomic DNA at the end. So the basic rules that you have to follow during this DNA isolation is that for the blood, first you lyse the red blood cells with gentle detergent such as Triton X100. You use your Triton X100 as a detergent in order to lyse your cells. And then for the hemoglobin, you can also inhibit restriction enzymes and tag polymerase. You can use wash them with that too, and you have to work on the ice always so that enzymatic processes slow down. Then further, you wear your gloves to protect your samples from us actually, because our small hair follicles on our hands, they're always danger of RNA contamination. And whatever the things, solutions that you're using, store them in the fridge and autoclave them at 121 degrees Celsius. And beyond that, keep all pellets and supernatants until you have the DNA you want. So some basic rules. So now to get into the DNA, you need following things in your. So these protocols of isolation of DNA: SDS page, DNA microarray, chromatin immunoprecipitations, cloning, you know, whatever the technique that you will do in your life, in your lab, with your colleagues, or you will teach to anyone. So this is my perspective that I have learned, that I love to cook actually. Mostly like to cook for my family, and if I'm cooking, so it's always there is a recipe that what ingredients it has to be added, what kind of things it's need to be added in a perfect amount at the right time in order to get the best results. If I add while making curry, Indian curry, let's say I add first tomatoes, then I add the vegetables, then I am adding onions, then I'm adding oil, then I'm adding salt. So everything is haphazard. Things are not the way it should be. So if I put everything like this in the pressure cooker and then just give a whistle to it, of course things will be cooked, but it will not cook the way it should be. First you have to make masala properly, fried onions, and then you add your spices, then on top you add tomatoes, then you add a little bit water, then let it cook, then you add your vegetables, then you mix them together, then you cook it. So things will work in that way. The same way with any protocol that you're working, you will see that it has to be have the exact amount of one ingredient has to be added, not more or less. If you do, your whole experiment is kaput, is not working. So it's the same way, like, so in the for getting the DNA, you use NaCl, which makes your DNA stabilized and keep it as a double helix. Then you use EDTA, which act as a chelating agent, as a magnesium ions, and as a cofactor of DNAs which chew up the DNA rapidly. So it just cut out the DNA. Then anionic detergent SDS is used, which disrupt the lipid membrane layers, which helps to dissolve membranes and your positive charges of chromosomal proteins, that is your histones, release the DNA into the solutions. Then you use your protease or proteinase enzyme to digest your proteins. So because we are isolating DNA, so we don't want protein to be present in that. So that's why we use proteinase K enzyme, and we incubate for the elevated temperature for 56 degrees Celsius to inhibit the degradation by DNases, and for 4 to 24 hours. It depends how you want to get it done. So these are the things that the scientists eventually, when they were doing experiments, they want to have pure DNA at the end. They get to know about that, if I add this will help me to have the pure DNA at the end. So that's why these things are essential. So these things, whatever the protocol that you're seeing in science, in molecular biology, it doesn't come in a single day. It's come with a lot of failures, with a lot of pain and a lot of frustration, and then you have the results at the end. So appreciate the people, even the person who have found out the PCR. It doesn't come in a single day. It's a lot of hard work behind that. So all the famous scientists who are working, let's say telomerase enzyme. So I just shared today on my WhatsApp status. So at the age of 23, she got the PhD. Carol Greider. States people are crazy, and 2009 she got the Nobel Prize winner in the physiology or medicine. Along with supervisors and co-laureate Elizabeth Blackburn, she discovered the enzyme telomerase, which is present over your chromosome, which helps in the production of a telomere. So today was her birthday. I just found out over the internet. Yeah. So this research, that you can see the papers on the left hand side in this picture, and that microscope with the various petri dishes inside, see Eppendorf on the right hand side, this magnetic stirrer. So yeah, this is the world of microbiologist, how things are working out. So respect each thing that you are working, respect, yeah, respect each thing that you are going through, you have learned within the field of biotechnology or molecular biology, because a lot of people hard work has been there. Okay, then getting rid of the protein. um, You can use organic solvent extractions using equal volume phenol is to chloroform of about 24 is to 1, and further protein at the interference of the centrifugations, so such as you can make it to the 10,000 rpm resolutions per minute at 10 degrees Celsius for 10 minutes. So in this way you can rid of the protein also. Then you precipitate the DNA by adding 95% ice cold ethanol of 2.5 to 3 volumes of DNA, and leave at minus 20 degrees Celsius overnight. And then you centrifuge for 10,000 P for 10 minutes, 4 degrees Celsius. Then you will see a pellet at the bottom. Then you air dry it, resuspend in the sterile distilled water, then store for the 4 degrees Celsius or frozen at minus 20 degrees is long term. So I mean, these protocols that you're seeing, it's very easy actually. There's a difference between a scientist. There's a difference between a scientist and, oh, there's working something. No, it's not. It's not working. So there's a difference between the scientist um, and technical assistant. So you will have technicians in the lab. Technicians can do anything if you tell them. They will repeat it and they will do it. Yeah. Whereas a scientist has to think why I'm doing that, what will happen if I do this way, and you have to analyze yourself. You have to think rationally that why you're doing something, what you will get out of it, how things will be working. So my one thing to work with you all students here is that you have to think rational also, that why we are adding that part into that. It's not like you have to learn these techniques by heart. Oh, I have to add this, I have to add in step 2 and step 3. I will give you this protocol, you go into the lab, you can do it anytime and you will get the results very easily. It's not a big deal. On the first day you will not get it, on the second day you will not get it, by the third or the fourth day you will surely get the results. So don't think that, because when I started my science, so I did a lot of mistakes on my in my first month. I was not even able to handle my pipette in my hand, not able to work in the chamber, laminar airflow. So but eventually you learn, you get better, and things get day by day confidence. You get the confidence that why things are not working, and eventually things works for you. So some basic rules we are done, this is done. Yeah, then you quantify them, your DNA at with a spectrophotometer with A260 optical density. Double stranded DNA will be 50 microgram per ml, single stranded will be 33. If it is RNA, then it will be 40 microgram per ml. And DNA purity at the 260 OD to 280 to 880 ratio, it should be between 1.6 to 2.0. If it is less than 1.6, then protein is contaminated. If it is more than 2.0, then it is a chloroform or phenol contaminated. Then you have to repurify your sample. So in summary, you have a sample for DNA extraction, you lyse your cells, you add your detergent enzymes, then you remove the cellular proteins, then you precipitate the nucleic acid with ethanol, then you quantify and purify purity measurement of your DNA. So that's it. Thank you very much. That's it for today. I hope I have shared this with you already, this workshop, or not. Yeah, and also you have to send me the assignments now. If I haven't shared with you this PPTs, so today we are done with the two parts. I think I have shared mainly the first one with you. Yeah, so I will share you this part also today. So send me the assignment of all the summary of it. And please do subscribe to my YouTube channel. That will really help me to grow. So yeah, it's with you now. Okay, then take care. Bye bye. If you have any question, just type me in. I will be happy to answer. By tomorrow. Those who can, it's great. Those who cannot, it's also fine. But if you can do it, I will really appreciate that. Really, really appreciate that. So we might have next class soon. So I will stop it here and we continue on the YouTube streaming itself. It has many advantages over other platform actually. Believe me. Cool then. Take care. Bye bye. Stay healthy. Stay immune. We have a lockdown in Delhi for 1 week, so we stay at home for next upcoming time. Ciao.